drug discovery is mechanism of manufacturing of drugs which are used to cure or prevent diseases. Now in drug discovery pipeline, target identification and optimization is a very important step. Now what is target discovery and what are its different perspectives and components? इस मॉड्यूल में हम ये देखेंगे कि टारगेट डिस्कवरी की कौन सी डिफरेंट स्ट्रेटजीज हैं एंड व्हाट आर देयर ब्रॉडर कैटेगरीज. नाउ number one thing is there is a great or important saga between target discovery and drug discovery itself. So that saga is basically, it is said that whenever there is a target basically when we want to make a drug, we say that okay, which parts or which components will be used for manufacturing of drugs. So once we define those targets, then the time between identification of those targets and manufacture of drugs and reaching in the market, which is called distribution, it is very huge. And huge means that it is roughly from 5 to 10 years. And the other thing is that at most, if there are 1,000 drugs are being made, only few of those make to the market. So that is basically distribution saga or target discovery to application or clinical application saga. Now when we talk about target discovery strategies, there are two broader strategies. Number one are which are specific to some disease and the other one are specific to some target. So now what are those specific to target and what are those which are specific to some disease? Disease centric or disease specific drugs are also called physiology based approaches. Physiology means which are related to normal functioning of a living organism. Opposite of this physiology based approaches, we have pathology based approaches in which the body of organism is not working in normal condition, rather it has some disease attached to that organism. So what happens in physiology based approaches? Number one thing is they are focused or based on disease centric approaches. Then there are multiple targets available. There is not a single target which they are trying to capture. There are multiple targets. Third thing is testing of these targets, it is done in vivo screening. In vivo screening means that within a living organism. A living organism, for example, if a drug is being made by for a human, then it will be tested on some other animal first, such as mouse or some other animals. So another thing is they are being tested by using different products such as small interference RNA or antisense oligonucleotide compounds. Moving on, as we said that this process it relies only on diseases, not on the targets. So in this case, target is basically a black box. Black box means that we don't know that what is the inner structure of the target. We are just focusing on the disease. As shown in this figure, we have a drug which is given to some animal or some organ system or organ part. Then we see what are its physiological effects. So physiological effects are again those effects which are related to some normal functioning of the organism. So there is no concept of direct target in this area. To remedy this thing, what we move towards target centric approaches. Now in these target centric approaches, as opposite to the disease centric approaches, we have multiple targets available and then we have to decide the best target that can be useful. So for any target, as you know that this target is basically genes and then the combination of genes with their protein products. So for targets, different targets, we need genes plus their protein targets, and then we need a target which produces the best molecular mechanisms. So now what happens is that because of different tools and advancements in biomedical sciences such as Human Genome Project and then different high throughput mechanisms such as high throughput screening, virtual screening, NMR methods, there are very good chances of many targets which can be tested at the same go. So because of these methods, it is possible that we make a target which is based on several targets, and then we select the best target. Another advantage of these techniques are they can be tested in vitro mechanism. In vitro mechanism means that instead of directly putting or directly checking some medicine or drugs on a human or animal, they can be tested in a laboratory. So in vitro, basically it is a technique. Vitro of course means glass, and these are the techniques which are tested in some laboratory settings. So they are not directly applied to animals or humans. Again, target centric approaches, they can be classified into two approaches. First approach is basically genetic based approaches, and the other one is mechanistic based approaches. Genetic based approaches, as the name suggests, they are directly related to genes and genome information. So based on genes and genomes, we decide, okay, which target is the best and which targets can be discovered or applied for medicines. And the other one is mechanistic targets. So mechanistic targets, basically, although they are also based on genes, but then they are focusing on physical nature of those genes, which are basically our mechanistic targets, such as receptors, enzymes, and in some cases genes also. But the good thing about this technique, which is target centric approach, is as shown in the figure, we have a drug, then we have target genes and pathways. So instead of in previous case where genes were black box, here we know what is the genetic structure, and then we have a drugs, and finally we get some molecular mechanism or genetic information which is used for drugs in future perspectives.